Hello, I'm Hannah Donner with the Collaborative on Health and the Environment. Change is bringing you the latest environmental health science through our partnerships, calls, webinars, science serves, publications, and social media. I would like to welcome everyone to today's CHE EDC Strategies Partnership webinar, which is titled PFAS Exposure and Epigenetics in the Firefighter Cancer Cohort Study. Our moderator today is Cheryl Patton, Director of Commonweal's Biomonitoring Resource Center. We will leave time following the presentation for a brief Q&A session. You may type in questions to the Q&A feature available on the menu bar at the top of your window at any point during the presentation. After the presentation, our moderator will read out questions for our speakers to respond to. We will get to as many comments and questions as we can during the Q&A period, and we'll follow up as we are able. For those who have called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speakers. This webinar is scheduled to last for 45 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Cheryl.
Welcome, everyone. And welcome to this very exciting webinar. I'm so pleased to facilitate. As you all know, firefighters are at increased risk for many cancers compared to the general public. And this increased risk is likely due to the exposures to carcinogens and other toxic chemicals firefighters may be exposed to on the fire ground, possibly in firefighter gear, and possibly in some of the firefighting tools currently in use. Today's presentation will focus on the mechanisms resulting from exposures to PFAS, which is per and polyfluoroalkyl substances, which could alter the expressions of genes that they then result in occurrence of some cancers, as well as decreased lifespan and aberrations in the immune system. Our speakers today are Dr. Jeff Burgess, who is a professor at the University of Arizona Zuckerman School of Public Health. His research primarily focuses on evaluation and prevention of injurious occupational environmental exposures. His research is funded by FEMA, NIHS, CDC, and NIOSH, and he's been long recognized as a leader in research that continues to inform how the occupation of firefighter can be made safer, which is particularly important given that the rural, wildland, and urban interface fires are on the increase across the country, and firefighting is very hard to make it safe. And he's been a strong advocate in helping firefighters maintain their jobs in a very safe way. Our second speaker is Dr. Jackie Goodrich, is a research assistant professor in the Department of Environmental Health Sciences at the University of Michigan School of Public Health. Her research program aims to identify environmental factors that contribute to disease susceptibility in vulnerable populations. This research includes investigation of epigenetics as a potential mechanism linking exposures to adverse health outcomes. And with that, I believe, Jackie, we're going to begin with you.
Yes, thank you so much, Cheryl and Hannah, for having us. Jeff and I are pleased to be here today to talk to you about this important subtopic and some research that we recently published in this area. So as a quick overview, Jeff will be talking to you about the general health risks from firefighting and the study that he's designed to look at some of these risks and to help protect firefighters. And then I'll talk to you about PFAS exposure that we've measured in a group of our cohort and how it's linked to epigenetic biomarkers. And if you don't know what that is, don't worry, I'll explain what that means when we get there.
Cool. So before I start, I just realized it might be useful to let everybody know how we met. I won't take too long time on this, but there was an administrative supplement to the NIEHS grants for our tox centers at University of Arizona and University of Michigan. And because of that, Jackie and I started working together on PFAS exposures and epigenetics, and it's been an incredible partnership. And so I've really appreciated the opportunity to work with her, and she's brought so much to the study of firefighters. So with that said, I'll go ahead, and I think you all are well aware that firefighters have lots of different exposures. So, you know, we think about smoke because that's what they often respond to, but there's many other exposures. And I think you have to, when you look at any health outcome in firefighters, you have to think about all these other exposures that are occurring. And, you know, with the realization that while you may be looking at specific exposures, you're not going to be able to measure all of the exposures that are occurring. So as stated here, there's heat, there's stress, which itself has some adverse effects. The PFAS, which we'll talk about today, particularly in aqueous film-forming foam, but other sources as well. PAHs, or polycyclic aromatic hydrocarbons, are always there whenever you have any combustion. There's many other chemicals, however, that also come out from burning buildings. And, we just measure the pHs because they're generally always there and we know how to do it, but there's a lot more to it. And then, of course, there's shift work, there's diet issues, et cetera, that all have health effects on the firefighters. And then furthermore, when you think about firefighters, we just think about the general municipal firefighters usually. I mean, those are the ones we see on the news. And now more lately, the wildland or wildland urban interface, or WUI firefighters, but there's many other groups of firefighters too. So there's aircraft rescue and firefighting, that's ARF. Those are airport firefighters by and large. They have particularly had exposures to AFFF. There's also trainers who get really heavy exposures during the training. And then there's fire cause investigators that often go in after the firefighters are done putting out the fire to figure out why the fire started. So these are all unique exposures. Next slide, please. So just a little bit about the Firefighter Cancer Cohort Study. This is a prospective multi-center study focused on carcinogenic exposures and effects in firefighters. And a good way to think about it is it's like a Framingham study for firefighter cancer, because we know that there's cancer. We don't know what's causing it necessarily and what to do about it. So just like with the Framingham study on cardiovascular disease, we hope to understand better what's leading to cancer and what we can do about it in firefighters. So the components are, we enroll firefighters, they fill out surveys about occupational and other exposures, collect biological samples, particularly blood and urine. The cohort itself is funded by a number of grants, which are more limited in scope. A lot of these are funded by FEMA. They're about 3-year grants. I'm just going to hit a few. This pre-FFCCS was actually work that we were doing in Tucson, and much of the research that Jack and I have worked on in the past really was involving just these Tucson firefighters, but it's expanded since then. The framework grant established, how we were going to actually do this, and with the uniform surveys across multiple fire departments across the country and multiple academic institutions such as the University of Miami and more recently Rutgers. Also NIOS is working with us. The expansion grant looked at certain different types of firefighters, such as trainers and investigators. We're going to talk about PFAS today, but we also have other studies on wildland urban interface, women firefighters, volunteers. That PACES grant is actually looking at PFAS exposures and COVID-19 risk, both from the actual infection and also vaccination. And then we just started a grant with wildland firefighters. Next slide, please. So this is just, an idea of some of the research concepts. Really, we're not just following people through to get cancer. I mean, we are monitoring that as an endpoint, but the whole idea of the FFCCS is prevention. So we want to do that through understanding and reducing exposures, through evaluating what the effects are and seeing if we couldn't prevent or potentially reverse that. And then even through to early detection of cancer, which we haven't done yet, but that is something that is a goal of ours. So you can see all the exposures in black here that we're already measuring. The ones in red that we would like to, that there have been proposals in, perhaps they haven't been funded yet, or they will be going in. And I also point out that in addition to cancer as an outcome, we also are starting to look at reproductive outcomes as well. And I think that's really important because a lot of the things that cause cancer can also cause adverse reproductive outcomes. And obviously there's, PFAS could be put into that category as well. Next slide, please. So just, again, I think all of you have probably heard about PFAS, per and polyfluoroalkyl substances. There's thousands of these chemicals out there, and kind of a word of warning, we're only able to measure a limited number of them in targeted assays now. I mean, there's a huge need to add additional ones. The targeted ones we're looking at now usually are the legacy chemicals. They have longer duration, elimination, half-life, so they'll stay in your body longer. Some of these other chemicals may come out sooner, but we don't understand the toxicity of many of these fully, even the legacy ones. This is not just a problem for firefighters. Again, I think all of you know that there's been groundwater contamination, throughout the nation in areas that we're using this PFAS containing aqueous film forming foam, or AFFF, and a lot of people drink from those water systems. And we also have it in cosmetic products, et cetera. So everyone is exposed to at least some of these PFAS chemicals. The toxicity you mentioned associated with some cancers, particularly urinary tract cancers, including prostate in that, and kidney, and others as well. Also a number of other effects, particularly immunologic mediated conditions. And then from the firefighter perspective, AFFF is only one source of exposure. There's also PFAS in gear, but we do not understand currently the extent to which it may get out of the gear into people, either while during use or from dust coming off of the gear, and then exposure through that as well. And then there's also likely some PFAS released from burning structures, but we again don't know the extent to which that is occurring. Next slide, please. So for the PFAS research we've done before, we're going to be talking with you today about work with about 200 structural firefighters from about 4 different departments in the country. We also have separate work where we've been looking at comparing incumbent and new recruit firefighters within one department. That's actually the Tucson Fire Department, and actually those new recruit firefighters over time. So that has not been published yet, but it's really helping us understand the extent to which firefighters are exposed to PFAS and how that changes over time. Jack and I will be working with Epigenetics on our firefighters, again, the airport firefighters in the future. The PFAS has been analyzed there. We just need to do the analyses. And then we also, in addition to the epigenetic changes that Jackie is doing, we're working with NIOSH to understand the in vitro toxicity using a number of human-derived cell lines. Next slide, please. So just so you get an idea, and actually if you can click the next one, please, Jackie, there. These are the standard legacy PFAS that are analyzed by the NCH laboratories at the CDC, where these analyses were done. The departments are A through D, and then the NHANES, or the general population comparison. And you can see across these legacy PFAS that everyone tends to have, well, for many of these, everyone has some extent of the chemical in their bodies. You can see on the graph here. Certain PFAS tend to be elevated in firefighters if they are elevated. And those tend to be either the straight chain or the sum of isomers for the PFOS, and also PFHXS. So again, some but not all fire departments, on average, will have an increase of these, but it's not every PFAS chemical. It tends to be these when we see it. Although some other department studies, including in California, have found other PFAS that are elevated and not these. So it does vary by region. Next slide, please. And just to give you an idea here, again, we're not going to talk about this group today, but when we look at airport firefighters, we believe that it's likely that there'll be some increased exposure, as we mentioned. And you can see here, particularly from this airport A, that had a limited number of firefighters in it, but quite elevated levels, again, of these three, the straight chain sum of isomers for PFOS, and then PFHXS compared to the NHANES, which is that last group in kind of a pinkish color. All right, next slide, please. So we actually have been reporting back this information to firefighters, and I think that's really important. The FFCCS is a collaborative research project, so we want to provide information back to the firefighters. We don't show the epigenetics results to them individually because there's so many of them, and Jackie will talk about that. But we do provide the PFAS levels back and compare both individuals within that fire department as well as in Haines. Again, without personal identifiers, obviously, for the other folks in the department. Next slide, please. And I will pass it on to Jackie at this point.
Thanks, Jeff, for that great overview. And now that you know why we're studying firefighters and what the framework for that is, I'm going to talk to you about maybe an unusual outcome that we study, which is, not a clinical disease, but instead a biomarker of toxicity. And so in case, some of you may work with different kinds of biomarkers and some of the audience might not. So I just like to define biomarker for you. We think of it as a chemical or the product of a chemical that's reacting with something endogenous, something from your cells that we can measure in samples taken from the human body. And the reason some of these things are so important is because we know exposures like PFAS can, perhaps cause cancer and other diseases, but if this occurs, it's not going to happen for years and probably even more likely decades. And so what's going on in between, clearly, the body is changing and leading up to this process of carcinogenesis that eventually becomes disease. And we don't want to wait until it's too late to screen that. So there are various kind of cellular changes we might be able to screen of things that are subtly changing in the meantime in that latency period. And as far as biomarkers go, we use biomarkers in lots of ways. We actually use them to estimate how much exposure you had by measuring things like PFAS in serum samples or PAHs in urine samples, and that gives us an indicator of not exactly the total amount in your body, but a good representation of what is in a person. And then we can also use blood samples to measure lots of biomarkers of effect or toxicity, some of these subtle changes that are happening, of which epigenetics is one that I'll be talking about today. And then ultimately, we think that those changes in between are, the susceptibility factor that might lead people to developing disease later on. And we hope in the future too to develop, prevention kind of screening tools, ways to use these biomarkers so we can help people who are at higher risk for disease. And in the case of the work that we're doing, we screen epigenetic biomarkers specifically. And literally the word epigenome means on top of the genome. And what it means in a more functional matter are changes in gene function or expression that occur without changing the sequence of nuclear DNA. So these aren't mutations that affect your ACGT code, but instead different chemical modifications on top of the genome that affect how it's packaged, where genes can be expressed and where they can't be expressed. And there's actually lots of different kinds of epigenetic modifications. The one we'll talk about today is DNA methylation, but there are other types too that you're welcome to ask about if you're interested. And I like to use metaphors to explain epigenetics for people that haven't really heard of it before. And my favorite is a music metaphor. You can think of your genome or genetics as a keyboard. If you come across a keyboard or piano, you expect it has the same keys, you know what notes they are, and that's like your genome. In each one of your cells, you have the same genetic code in each cell, and that's pretty reliable. You know it's there. But even though you have this one code, it can be used in many, many different ways. And that's like the epigenome. You can use a piano to play, millions of different songs and express it in different ways. And in the same way, your genome is expressed in different ways in healthy cells versus disease cells, in, an embryo versus middle-aged, liver cell. Everything is just, every cell has its own function. And even though it has the same genetic code, and it's the epigenome that is programming that, deciding what genes are turned on and off and when. And why, before we get into why we look at this in environmental health, a little bit more about DNA methylation, because that's the part of the epigenetic code we'll be talking about today. It's really actually quite a simple modification. In this cartoon, you can see that there's this little M on top of the C's, and that's representing that in mammals, methylation almost exclusively happens on cytosines that are next to guanines. We call them CPG sites. And typically, not always, but for the large part, if you have a lot of these methylated CGs, especially in the promoter region of a gene, it'll block transcription factors from binding and that gene will be turned off, though it is a little bit context specific. But for those of you who like biochemistry, it's actually a very simple modification. This is a usual cytosine. When it's methylated, the only difference is this methyl group here replaces this hydrogen here. So it's a very small change. But on the broad scale, you know, if you have a lot of it or if you don't have much of it in a certain region, that can have big impacts on how genes are turned on and off and expressed. But why do we want to measure this in firefighters? So we already talked about how firefighters have increased risk for cancers and how they have a lot of exposures, including to PFAS, that could be contributing to this risk. But there's also this big connection that we know now from the last 10 or 20 years of research on how exposures can impact the epigenome. And there's been, you know, studies in various populations, not many occupational yet, but lots of other populations on lots of different exposures, from chemicals to things in diet to even things like psychosocial stressors. And it's been shown and suggested that these exposures can modify the epigenome. And why is this a problem? So here in this little cartoon, this is DNA with some methyl groups on it. Say you're exposed to some PFAS in your environment or from your occupation, and it leads to the loss of some methyl groups at this gene. You know, this isn't just a problem for the one cell that was affected. The thing about epigenetics is that it can be propagated every time a cell divides. So now, after this exposure happens and after this change happens, it might be replicated in various cells that come from, you know, this original cell. And if it's impacting a gene that's important for some function that you need, then eventually all of that could contribute to disease. And so that's, again, one of the reasons why we're measuring epigenetics, because we know some of these exposures they have could change the epigenome. And we also know from other studies that I won't get into today that cancers have lots and lots of huge epigenetic changes. So we know that epigenetics is a big part of the processes that lead to cancer. And then we also know that the epigenome could change before disease occurs. So it's one of those biomarkers in between when you have an exposure and when you have a disease. And, you know, and we could use it not only for risk assessment, but also to possibly, you know, develop into biomarkers to help decide who's at high risk for disease and who's not, and help the people that need the most help there. Now, coming back to, I'll tell you about two specific research questions with the remainder of our time in this area here that I worked on with Jeff and the FFCCS. And our first question was, do exposures to PFAS specifically accelerate epigenetic age in the firefighters that are in the cohort? And I haven't really touched on this yet, so I'll tell you what epigenetic age is in a second. But as far as methods go, this is just for the people that want some details. But the way we do our DNA methylation analysis is actually use this array technology that can quantify DNA methylation at specific sites, in 850,000 of them. So they cover all throughout the entire genome. There's some in every known gene. And we can also use these data to get specific methylation at specific genes, and also to calculate what we call epigenetic clocks. And then we use these data, this big data, to answer our various questions. But what is epigenetic age? So this is yet again another biomarker, but this time it combines epigenetics, but also gives you an indication of biological age. So it's known from various research that DNA methylation at certain regions of the genome is strongly predicted of your chronological age. And this knowledge has, with data from thousands of people, has been used to develop these clocks that are based on certain parts of the genome that can approximate your biological age. And these are just some of, let's see, there's 5 here. We use 2 more that aren't in this graph, but there's different ones. They're all kind of related, but each a little bit different. And we calculated these clocks in our cohort because what's interesting about it is if you are aging in a sense faster than you should be, then your epigenetic age will look older than your chronological age. And also the reverse is true too. And why these are important, you know, for one, because, you know, it's not really a good thing to have an accelerated biological age, but in addition to intuitively, you can think about that, but also accelerated age at these clocks as listed here in big cohorts has been linked to cancers or cardiovascular diseases or all-cause mortality. So in some ways, it's a predictor of adverse health outcomes too. And our question was to see if the PFAS exposures were linked to accelerated epigenetic age. And we did this and the rest of our study in a group of about 200 municipal firefighters from the Firefighter Cancer Cohort Study. So all of these individuals had their PFAS exposure assessment and their DNA methylation analysis done, and they came from 3 different departments from different places in the US. You can see that they were mostly male and white non-Hispanic individuals, and they were, about 38 years of age on average. And they were, in general, this is a healthy cohort. And for those of you who are interested in, who have looked at PFAS numbers before, you can see on the left the PFAS analytes that we measured in their samples. And an important column to look at is the percent above the limit of detection. And you can see that there's only 3 PFAS that weren't detected in that many people. The rest of them were essentially detected in everybody, and there was wide ranges of values measured amongst individuals too. And so let's get to some of the results. This is compiling a lot of results in sort of a simplistic manner, but the take-home point here is if you see something highlighted in blue and it's bolded, that's a statistically significant relationship between that PFAS on here and the clocks going across the top. And you can see, and this is after adjusting for confounders and things like that in these 200 people. And you can see that, for instance, PFOA was associated with accelerated aging at almost all of the clocks. And then other legacy PFAS, PFHXS, and the sum of the isomers for PFOS were also associated with a couple of clocks. And then some of the longer chain PFAS weren't really significantly associated with anything except for an inverse, which is an unexpected direction association with 1 clock. But we thought the big take home from this part of the analysis was that these 3 more legacy PFAS were associated with accelerated aging, at most of the clocks, which is, not the kind of relationship you want to see. And so moving beyond that, and in addition to these clocks, we have data at every known gene, and we want to make use of that too. And we asked the question of, are PFAS exposures associated with DNA methylation at specific genes, and which ones? And we hypothesized that the genes would be in pathways relevant to cancer or possibly immune function or other disease states that have been linked to PFAS exposures in the past. And we used the same group of people and did lots of models, since we have 9 PFAS and 850,000, or I guess after screening some out, 740,000 CPG sites that passed all of our quality control checks. And when you do this many models, you want to adjust for multiple comparisons, because you could just come up with a lot of false positives just because you're running so many models. And so we used a very strict p-value cutoff, and which means the genes that we picked up are probably the very strongest associations. There might be some that we didn't pick up. And you can see here that several of the PFAS, including some of the legacy and some of the longer chain ones, were associated with DNA methylation at specific genes, or at PFNA was associated with methylation at lots of regions, which is when we combine data on sites that are next to each other. And when we tried to look at, what are those genes, what's interesting about them, what pathways are they in, and we published this in Epigenomics. So if you're an epigenetics person, you can read the paper and look more at read about more details about these genes. But giving a brief description, a lot of the genes that we found with significant regions or sites are genes that are involved in processes related to cancer. Some of them are listed here, and there are also a couple that do have known functions in the immune system too. And in addition to that, we did this pathway analysis where you can load in all the top genes and see what pathways they're linked to. And I know there's a lot listed here, but some of the take-home points here are that some of the pathways for PFAS, for instance, at the top, are related to, lipid regulation of fatty acid transport and synthesis and things like that. And we know that PFAS have been known to affect cholesterol levels and other lipid pathways. So that makes sense with what we know. There are also a couple of pathways here that are important for cancer, such as Hippo signaling. And for PFNA and PFDA, there were a lot of pathways that had to do with cell migration and ion transport and things like that. And these are important cellular functions for many processes. They can be important for cancer, but for other states as well. And in conclusion, out of all the data, that we saw from this project, I think the main conclusion is that we saw evidence for a link between some of the legacy PFAS and accelerated epigenetic aging, which we want to follow up on in other firefighters in our study beyond what we show here. And then we also saw evidence for altered methylation at key genes that may be in pathways important for cancer development and immune function and other disease outcomes from several of the PFAS. And one important thing to note is that this was a cross-sectional study, so we can't prove causality with these associations. And it's also possible, especially based on the exposure data Jeff showed, that some of their exposures are not just from the occupation, it's also from the environment, from various sources in the environment. So that means a lot of these results could be applicable to the general population too, but we do want to do some more digging to figure out with some more longitudinal studies to see what the contribution of specifically occupational exposure is as well within these associations. And just to summarize too, why we want to use epigenetic biomarkers, hopefully this kind of research can help to inform risk assessment of hazardous substances to the public or to occupational groups. It can help us to understand some of the subtle mechanisms of toxicity that lead up to disease. And that could help us to develop prevention and intervention strategies to protect health or to promote health among people who are at higher risk. And the long view of the broader cohort study are to identify the most harmful exposures, PFAS, but also others that can be targeted for prevention and reduction to protect firefighters, and also to identify biomarkers that can warn us of risks early on, and of course to inform intervention strategies to protect health and possibly occupational health policy and things like that. And this work is done with a huge team. Listed here are the people that worked on the specific paper, but there's a lot of people that work on the other interrelated studies that are part of this. The main funding for this one was from NIEHS, NIOSH, and FEMA. And we have, a big research team, including our very important fire service partners, without whom we wouldn't have a cohort at all. And with that, we will both answer the questions that you have.
Okay, let's go right to the questions. We have several good questions for you both, and I'll read them out. Most of them are from Tommy Kajir, who's a Danish Firefighter Cancer Society. So his question has to do with how you might, he basically is talking about in Europe, PFAS was banned in 2011 for use in firefighting foam. And in Denmark, the use has been pretty much stops already 2005, 2008. So the pollution from PFAS, he feels, comes mostly from the water they drink and other food products. So they do see high concentrations of PFAS of citizens living in specific areas which are much higher than concentrations in firefighters. So looking at blood tests from firefighters today seems problematic in order to link it to firefighter foam from 15 years ago. So is it possible at all to look at firefighter blood samples from 15 years ago to kind of get a sense whether PFAS is a primary source of exposures to firefighters?
So I'll take a first stab on this and then Jackie can add on. But Tommy, thank you for the question. The whole point of the Firefighter Cancer Cohort Study is, we want it to go on for 30 years or more, given that latency period that Jackie was talking about between exposure and onset of cancer, which can vary, as short as, a few years, maybe 3 or so for some forms of leukemia and over 30 years for some forms of lung cancer. But, we only started the putting people into or enrolling firefighters in the study back in maybe about 2015 or 2016. So we don't have that biological samples from before. So while that could certainly be done and see what the exposures are, we, in firefighters previously, we don't have access to that. The other thing is, since, a number of these PFAS, like the certain legacy ones like PFOA, PFOS, have been banned from further production use in the US and other countries, that you actually can see the levels going down in the general population over time. So it is reducing for most of these legacy PFAS over time in those studies that look at representative samples of the population like NHANES. So you would actually expect to see maybe higher levels since these things were started to be used in like the 40s and the 50s. You might actually see decreasing over time.
Okay, another question from Sarah Howard, who says an excellent presentation, as I'm sure most of our participants would agree. She's wondering, do cancers and firefighters vary by job type? And possibly, as you mentioned, there are several sources of exposures to PFAS. One might be fast food containers, which may be still aligned with PFAS, and firefighters on the fire ground might be eating a lot of fast food. Would that be one thing? And the third part of this is, are you any chance of looking at the offspring of firefighters to see whether the aberrations in the gene expression might be inherited? If those exposed, is that inherited to the offspring of firefighters?
Well, go ahead, Jackie.
Here, Jeff. Well, I was going to say, Jeff, you can do the first, and I'll do the second question there.
Sure. So we actually plan to do job titles within firefighters to see if there's association with PFAS. And some of these same individuals that Jackie had described of the structural firefighters, and a few more I think that we've added since. So that's underway. I believe in some of the other studies that have been done, including I think in California, they may have found some differences by role, but you also have to realize that some firefighters change roles over time too. So kind of saying what they are at this point in time may not reflect what they were doing before, but that is an area of kind of active research. But I think what we're seeing more is more regional variation, I think, so far. And again, that might suggest that there's a background environmental exposure that's going on, or perhaps different practices within the fire departments.
Yeah, and for the second question, I know I personally would love to look at the offspring of firefighters. A lot of the work that I do outside of the occupational realm is looking at mother-child pairs and the effect of various exposures on children's epigenome and outcomes throughout their lifespan. I think at this point, this isn't something that we have funded right now, but it's something that various people that we work with have been interested in and have been trying to get going. So hopefully this will be a direction that we can go in the future. There is one small study going on right now that does have the ability to look at sperm and some measures there, which will start to get at some of these questions.
Okay, thanks for that. And then there's a question about the returning of the results. What kind of information do you provide to the firefighters about how they can reduce their exposure, and have the return of results process been well received? Are firefighters alarmed, or do they say that the exposure is too intense, not too much can be done? What kind of feedback are you getting back from firefighters and the results conveyance?
So we, as Jackie said, work very closely with our fire service partners. So on every project, we have the fire service research team members that give us feedback. So we were very careful that when firefighters were enrolled in the study, that we explained to them what we would get back and what we'd be able to do. And we emphasized that during the report back session. So we, as I showed you, had that one slide that kind of showed people where they were relative to others. We provided an ATSDR fact sheet that had some information on PFAS and health effects, but we said to them, we cannot tell you what the health effects will be associated with this. The best we're going to be able to do is just to show you where you are compared to NHANES, the general population, and other firefighters in your department. And when we were enrolling people, we told them this, they understood that. I think they appreciated knowing where they were. It's a form of documentation. And during these question and answer sessions, people had a chance. So it wasn't just a report back, but we had Q&A sessions as well, where people could ask questions. So we made sure that we gave every opportunity to ask questions. And we also would take individual emails and phone calls from our participants as well. So I think it went well, even though we could not tell them what the health consequences were. Now, the last bit about what you can do about it, we didn't, at that time, there wasn't much that you could do except, as I think pointed out in the ATS-JAR document, is to avoid things that are known to have higher levels of PFAS. For example, some forms of dental floss are known to have increased, PFAS chemicals. And if you could check your water system and know whether there was PFAS in your water system, and you could avoid that if you wanted to. Many water systems, municipal water systems, report it back. Subsequent to that point, there is some evidence that I think there's been one thing published, but just in a general sense, and then very specific study out of Australia that hasn't been published yet, where they're showing that if you give more frequent blood donations or plasma donations, you can actually drop your levels of some PFAS chemicals. And it's actually interesting to see that women in general have lower levels than men, and that may be due to menstruation.
Okay. Regarding the serum levels of airport firefighters, do you have serum data for airport employees that are not firefighters?
No, we do not. I think that would be a very interesting question, but the projects by and large are funded for the firefighters, and for that reason, and I think just challenges in getting this comparison of non-firefighter populations, that hasn't been something we've been able to do so far.
Okay. And there are questions about the possibility of a dermal uptake through the skin, and there's some research on that. Can you comment on that a little bit? You mentioned a little bit during the slide presentation, I believe.
So the best work that I'm hearing about now is from our NIOSH colleagues. They're not on our project specifically, but within NIOSH, there's an individual, and I've forgotten her name, unfortunately, that's actually looking at that in an animal model and finding some degree of dermal exposure or penetration, absorption. But in general, I think for most people, the source of exposure will be ingestion.
Okay. And then another question is, PFAS, through an inhalation or ingestion, goes into the bloodstream. So, and then from the bloodstream, PFAS has been found in different organs in the body, including the brain, the lungs, the heart, bones, and so on. So if PFAS is removed in the process of blood donations, would the PFAS in these organs be taken up by the serum or the blood itself as it circulates through the body? Is there any kind of transfer that happens? Do you know anything about that?
I don't have great knowledge on that, but just based on medical toxicology principles in general, where you have these deeper compartments, if you do remove it out of the circulation, there is some equilibration where the levels might they'll slowly leave that tissue and get back into the circulation because they are in equilibrium. And from what I know about these, again, the studies that hopefully will be published before too long, there is a reduction that they're seeing after a significant period of time in donating blood or plasma. So I think that's probably a true reduction in the amount that's in the body.
Okay. Okay. And then have you found anything unique in female firefighters as compared to the male firefighters in terms of epigenetic expression, cellular or pathway damage? Is there a difference between females and males?
Yes, that's a great question. And that's a big area of our ongoing research. As the person who asked the question, I see they could tell we only had, about 15 females in this group. But because of that, we're working with another collaborator, Sarah Jenke, who has an entirely female firefighter cohort that she's building and is kind of incorporating into the FFCCS. And so we'll be getting the sample soon. We haven't done the epigenetic analysis yet, but in the study I reported today, you know, there's not enough power at all to look in that small group of females to see what's going on only with them. But we will be able to do that because we're going to have several hundred female firefighters within the next, you know, year or two with their epigenetic analyses complete. And we will be looking at their data, which is very important because obviously there's sex differences and everything. But, you know, even within the studies that have been done of firefighters and cancer risk, you know, among female firefighters there have been increased risk of certain cancers, including, I think one of them was bladder cancer, was like 1,000 times higher or something compared to the general population. Granted, there's probably like a small sample.
I think it was 10 times higher, Jackie. I think 1,000% increase.
Oh, 1,000%.
Oh yeah, sorry.
10 times higher, sorry. But yeah, so we know that's very important to look at.
Anonymous attendee, can you discuss the difference on the cancer-related effects of linear PFAS and branched PFAS isomers that you see in your study? Is that something you took a look at?
Yeah, and so, you know, in our study, I think the person asking the question probably noticed that I think the branched PFAS isomers were associated with more methylation at specific genes and accelerated epigenetic age at more biomarkers. But there's a few possibilities here. You know, of course, linear and branched, because of their different structures, can have different effects, can have different reactivity in the body. That's one possibility. Another possibility is that we just didn't have, they're doing something similar, but we didn't really have the power necessarily to pick it up, because I did see a similar direction of effect for the linear PFAS, but it just didn't reach statistical significance. So this is something to continue to look out for in the rest of our study as we, you know, we're trying to get data on hundreds more people in the next couple of years. And this is also something that our in vitro collaborators do. You know, they look at the effects in cell models and things like that on individual PFAS and see how they act differently or the same.
Oh, we have many more questions, but we've reached the 45-minute limit that we presented. So I'm going to turn the webinar over to Hannah to continue on with questions because there are many more that perhaps we would like to see answered. But I want to let everybody know that if they need to leave the call now, that's possible. We'll continue on for a few more minutes. So Hannah, can I turn this over to you now, please?
Sure. Thank you, Cheryl. Yeah, so we had a couple questions just going on here. And then if we don't get to any questions, I'll pass them on to the speakers. And as they're able and would like to answer, they'll get back to you. But I just wanted to ask you, there's this one question that's really interesting. So it says there's been progress in establishing guidance for fire departments, that it's worth taking a look at these specific recommendations by Dr. Vasili. This information isn't always making it to the fire service members and families. And I think they wanted to know, this includes suggestions like using turnout gear for any purpose or other than fighting a fire. And they want to know, is there anyone studying the impacts of fire service families and also getting to this fire family, those firefighters' families outreach? How's that going?
So there, you know, we're not the only study, obviously, that are looking at PFAS, and so I've heard the same things about, you know, fire departments limiting the use of their turnout gear, right, to actual fire, you know, or other appropriate type of incidents. Again, we do not know the extent. We do know that there's PFAS in gear, at least in most of the gear that's used in the U.S. And I can't say in other countries, but we don't know if that PFAS is getting into the individual firefighters. So I just want to kind of emphasize that. However, taking a precautionary principle and not using, you know, your gear, your turnout gear in type of responses where it's not needed is very reasonable approach. From the perspective of, you know, the effects on families, Jackie had mentioned that, you know, we're looking at kind of the epigenetic route as potentially a means by which an exposure could affect the offspring of a firefighter, be it a male firefighter or a female firefighter. But you know, in terms of taking things home from, you know, the fire department, like if, you know, that generally they should be cleaning their gear at the fire department. They shouldn't be taking it home with them, because there's a lot of other stuff that we know is on the gear and can get into people, like PAHs, et cetera, that, you know, should be prevented. Also, Michigan, I think, is doing a pretty big study of firefighters, Jackie. I don't know if there's anything there that would be relevant to this question.
Yes, the state has recently launched a big PFAS surveillance effort, part of which is in the general population and part of which is in firefighters from throughout the state. At this point, though, they're not doing anything with their families right now, but I think they also have hopes of, you know, sharing out results and trying to make recommendations. And the state is actually, I believe, has banned, you know, the firefighter foams and has a take back process too, to get back any that are still stored at old fire departments. So I think they'll be in a position to look at, you know, ongoing sources of exposure, like gear and things like that.
And just so for everyone who's not completely familiar with the AFFF, the aqueous film forming foams, again, those are used for hydrocarbon fires, like, for example, at airports or petrochemical facilities, because they'll float on top of, you know, the hydrocarbons, fuels, and suffocate the fire. But that's why a lot of the, you know, the contaminations occurred at airport sites and at petrochemical sites. But not all AFFF has PFAS, just certain kinds, the mil-spec ones that have been required, you know, for use at airports until recently. But, you know, are ones where you always see PFAS, but otherwise you need to look at each PFAS individually to figure out, at each AFFF individually to figure out whether it has PFAS or not.
Great, thank you. There are some questions here on blood transfusions, and, you know, so if you can drop your blood levels by PFAS by donating blood for plasma, does it mean that it's passed on to the receiving transfusion? Receives the transfusion?
Well, yes, it doesn't really break down, or most PFAS at least. So yes, it could very well be in transfusions. And I'm wondering if, you know, that's something in the future that will be screened or monitored in some way.
Great question.
Yeah, and speaking of which, are you looking at other sorts of cancers like breast cancer in the future? Are you considering that? There's this one small study they referred to of 200 female firefighters that said they found an increase in breast cancer.
Yeah, I think you know, the nice thing about the way our study is structured is that, well, in many ways right now it's pre-cancer, you know, so we're recruiting people and hoping to follow them for, as Jeff mentioned, 30 years if possible. So with that in mind, I mean, we would capture data on any cancer that occurs in our cohort, including breast cancer. And the way the epigenetics works too, you know, the genes that we screen include all known genes, which means when we get the data on the female firefighters, I can dig into known breast cancer genes and see what those look like specifically and things like that.
Great. So I guess this is the big question that we all probably have, is just in terms of regrettable replacements or substitutes. What do you see, this is a big question, policy question, as being safer alternatives or of ways that we can work towards regulating PFAS as a class? And which, if not as a class, which PFAS, with your research and what you see, do you see really targeting to prioritize?
Jackie, did you want to start with that?
Yeah, I mean, I think it's very tricky as far as which I agree with the plans to regulate them as a class, because that many researchers in this field, including people who've been on this webinar before, have been making, because, you know, we measured here 9, but there's really 9,000. And we know these 9 are toxic, but that's not the only reason we measured them. It's because there's a good method to measure them. So that means there are other PFAS that could have very similar effects, but we just haven't been able to measure them in these big human studies yet. So we haven't looked at the effects yet. And I mean, I think there's a lot of good computational toxicology methods that can be used to infer which ones will act like each other. And that's really important because, you know, we don't have time to evaluate all of these PFAS individually. And Jeff can answer this part. I think the newer, supposed to be safer foams are supposed to be completely fluorine free, right? Or there's some
Well,
like that?
exactly. So, for example, at Heathrow and some other fire departments internationally, they have PFAS-free AFFF, but these aren't drop-in substitutes. So, you can't just put them into the same vehicles that we've got at our airports. They require, expensive new equipment. But this is an active area. We are actually looking at working with our NIOSH partners again, the toxicity of those fluorine-free foams, just to make sure that we're not, well, again, it's toxicity screening, as I think many of our participants in this call know, you have to figure out how broad you're going to go. I mean, there's many different tests you can do, but at least to start with some human cells and make sure that we're not seeing increased toxicity with some of these fluorine-free AFFF or other firefighter foams. And from a perspective of kind of gear, I mean, if we can find I think that it's not unreasonable to develop, and I think places are doing this, firefighter gear that doesn't have at least stain-resistant coatings on the outside. I think there's some other issues in terms of kind of like the vapor barrier inside that are complex. But I do think that if anything else is going to be added to the gear instead, it would be really important to make sure that that is also tested for toxicity.
Great. All right, one last question, then I'll let you guys go. Is there any suspected synergy between PFAS and toxicity?
I think maybe a better way to think about that question is there maybe synergy between PFAS when it comes to toxicity? So we know, or between PFAS and other chemicals firefighters are exposed to, like PAHs and things like that. And that's, another kind of new place our field is going. We used to just look at things one at a time. So we definitely want to, with the data that we're collecting, as we get it on more and more people, we'll have, better power and capabilities to look at the impact of all the PFAS, collectively. You got to use fancier statistics to do that, or to look at, PFAS and other exposures people in the fire service have. And, I'm sure we probably will find additive or, potentially interacting synergistic effects when we combine all of that data, but we need to do that still.
Great. All right. I'm going to go ahead and close up here, but wanted to thank you so much, Jackie and Jeff, for presenting today. We're approaching the end of today's webinar. A video recording will be available on SHEA's website soon, and tomorrow you'll receive an email containing a link to the video. The next SHEA Alaska Partnership webinar will take place on tomorrow, December 16th, and is titled The Challenge of PFAS with Dr. Linda Birnbaum. To learn more and to RSVP, please visit our website at healthandenvironment.org. If you are new to SHEA and would like to stay updated about upcoming events or more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website at healthandenvironment.org. Additionally, if you appreciate these SHEA partnership webinars bringing you the latest environmental health research for free, we encourage you to support SHEA's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, thank you so much, Dr. Burgess and Dr. Goodrich, for taking time today to present. And thank you to Charles, who's not here with us anymore, for her moderation. Thank you so much for joining us. Have a great day. Stay well and healthy.